Welcome to the deep dive. You know, we are tackling something today that, well, it honestly feels like science fiction.
It really does, yeah.
Because think about this for a second. If you extract a brain to map out all its wiring, you kill it.
Right. You preserve the structure, but the function is gone.
Exactly. But if you keep it alive to watch it, think, well, you can't see its intricate wiring. For decades, neuroscience has just been trapped by this fundamental trade-off.
It's the ultimate measurement problem, honestly.
Yeah, you're forced to choose between form and function. You can track a neuron's electrical firing pattern in a living animal, or you can euthanize the animal to, sequence that neuron's DNA and trace its anatomical shape.
Right, but you can't do both.
No, you can't. Trying to get the live behavior, a complete physical wiring map, and the underlying genetic blueprint from the exact same cell simultaneously has been biologically impossible.
It really has. I mean, to get high-resolution structural or molecular data, you have to fix the tissue. You use chemicals to freeze the biological machinery in place so it doesn't degrade.
Right, you lock it down.
Exactly. But the moment you fix the tissue, the system is dead. You lose all that dynamic information, the actual computation the brain was performing.
So you just end up guessing, right?
Pretty much. Neuroscientists have historically relied on statistical correlation. We measure function in one group of mice, anatomy in another, and genetics in a third group, and we hope the patterns align.
And hoping patterns align across entirely different samples is a very long way from actual ground truth, which is exactly why we are exploring this groundbreaking 2026 paper published in the journal Cell by Zhao and colleagues.
Yeah, they decided that statistical correlation just wasn't good enough anymore.
They really did. They targeted the primary visual cortex of the mouse, the VISP, and engineered this incredible pipeline to capture all three dimensions of data from the exact same individual neurons. They call it the Imaging-Based Multimodal Characterization Platform, or IMC.
The IMC platform it's essentially a masterclass in non-destructive sequential data harvesting.
It's like tracking someone's job, their daily commute, and their DNA all at the exact same time.
That's a great way to put it. To pull this off, the team had to figure out how to layer three completely different imaging technologies: live functional imaging, whole brain optical sectioning, and molecular spatial transcriptomics.
And they had to do it without the preparation for one ruining the tissue for the next, which is wild.
Yeah.
So let's break this down. Step 1, capturing the live function.
Right, the job.
Yes, the job. How do we actually watch a brain thinking in real time without scrambling it? They used in vivo calcium imaging, but they couldn't just light up the whole visual cortex, right?
No, definitely not. If every neuron fluoresces simultaneously, you just get this massive optical traffic jam. You can't isolate the single wires later.
So to solve that optical crowding, they used this dual virus sparse labeling strategy.
Right, they co-injected two viruses into these living, awake mice. One virus carried Cre recombinase, and the second carried a Cre-dependent fluorophore called GCAMP P6s, or 8 meters.
Okay, let's unpack that for you listening because it's a vital detail. Think of Cre recombinase as a biological AND gate.
Exactly, a logic gate.
The GCAMP fluorophore is the payload, but it remains locked and totally inactive unless the Cre enzyme is present to snip away a specific blocking sequence of DNA.
And because they injected the Cre virus at a very low titer, a very low concentration, only a random, sparse subset of neurons actually receives both components.
Which isolates our targets beautifully. We get these distinct glowing bulbs instead of a blinding floodlight. Now, GCAMP doesn't just glow randomly on its own.
No, it's a highly engineered protein. It fuses green fluorescent protein with a calcium-binding protein called calmodulin.
Right. So when a neuron fires an action potential, voltage-gated calcium channels open up, and calcium just floods into the cell.
And that calcium binds directly to the GCAMP. It forces a physical conformational change that suddenly allows the molecule to emit bright green light, but only when hit by a 2-photon microscope.
So that green flash is a direct proxy for an electrical thought. And to test this, the researchers basically did what I like to call the flashlight test.
The flashlight test, I like that.
Yeah, they placed these awake mice in front of screens and shined specific visual patterns at them. They used contrast reversing checkerboards, which test spatial frequency.
Right, and contrast sensitivity.
Yep. And they also used drifting sinusoidal gratings, which are just these moving parallel lines, to test motion and orientation tuning.
And crucially, they also tracked facial motion energy. Things like whisking and jaw movement.
Oh, right, because the mouse's arousal state actually modulates how the visual cortex fires.
Exactly. It's a very noisy environment, so they had to track the physical movement too.
And out of the thousands of neurons in that region, they successfully isolated and recorded the live firing patterns of 207 specific neurons.
And very clear physiological profiles emerged from those 207 cells.
Yeah, some neurons fired exclusively for the checkerboards. Others were tightly tuned to just the moving gratings. A third group responded to both. And a totally distinct subset only fired when the animal physically moved its face.
Which is incredible because now we have our baseline functional dictionary. We know exactly what specific computation those 207 individual cells are responsible for in the living animal.
Okay, let's pause here and lock this in for a second. We've got our first small recap. Bullet 1. We watched live neurons react to specific visuals using calcium imaging. That's the function.
We have a map of 207 distinct neural workers and their exact jobs.
Right. But a firing neuron is completely useless if we don't know who it's broadcasting that signal to. So here's a question for you to consider as we move to step 2. If you knew what a neuron responded to, like, say, moving lines versus static contrast, would you expect its physical outbound wiring to look entirely different?
Keep that in mind, because to answer that, we have to transition to the morphology phase, the commute.
The physical wires.
Exactly. The challenge here is tracing a single microscopic axon that might be millimeters long, winding through the incredibly dense neuropil of the entire brain.
But wait, you can't physically slice the brain to trace it, right? Because the moment you slice it with a blade, you sever the axon?
You cut the wire, you lose the map.
Okay, but if you can't slice the tissue, and you've injected a structural dye to fill those cells, how on earth do you see through a whole intact mouse brain? I mean, the brain is packed with lipids, fat scatters light, a whole brain is completely opaque.
And this is where chemical tissue clearing comes in. It's brilliant. Before extracting the brain, they inject a third virus to express MEGFP.
Which is a much stronger, more stable structural green dye.
Yes, it floods the entire neuron, filling even the furthest, most distal axonal branches. Then they extract the brain and immerse it in an organic solvent mixture called BABB.
That's BABB, right? Benzyl alcohol and benzoate.
That's the one.
But rather than just explaining what BABB is, we need to explain the physics of why it actually works to make a brain invisible.
Yeah, it's about refractive indices.
Exactly. When light passes from water into fat, it bends and scatters. That's why milk is white and opaque. It's water and fat mixing. BABB works by chemically stripping all those opaque lipids out of the brain tissue and replacing them with a fluid that has the exact same refractive index as the remaining proteins.
It essentially homogenizes the optical density of the tissue. It renders the entire brain as transparent as a block of glass.
It's like turning frosted glass into a clear window.
Precisely. And it perfectly preserves those glowing MEGFP-filled neurons suspended in this 3D transparent space.
But even with a perfectly transparent brain, scanning a 3D volume that large at synaptic resolution using a standard 2-photon microscope, that would take weeks, wouldn't it?
Oh, absolutely. The structural dye would photobleach and degrade long before you ever finished tracing the axon.
So how did they speed it up?
They custom-built a multiplane 2-photon microscope, or MPTPM.
Multiplane. So it's looking at multiple layers at once.
Yes. Instead of a single laser rapidly scanning point by point like a traditional setup, they use a holographic spatial light modulator. It alters the phase of the incoming laser, splitting a single beam into 8 distinct beamlets.
8 lasers at once.
Right, focused at 8 different depths simultaneously. So you get fast, high-resolution 3D scanning of the entire intact brain.
That optical multiplexing increases the scanning speed by nearly an order of magnitude. And using this, they traced the full brain-wide axonal arbors of those exact same 207 functionally recorded neurons.
And the anatomical routing they discovered was highly, highly specific. They separated the neurons into 2 well-known morphological classes.
The pyramidal tract, or PT neurons, and the intratelencephalic, or IT neurons. Let's define those.
Sure. We can think of IT neurons as the local peer-to-peer network. Their projections stay within the cortex and the striatum. They share information locally.
Right, they never leave the neighborhood.
Exactly. PT neurons, on the other hand, are the long-range broadcast network. Their axons plunge deep into the subcortical regions of the brain, sending visual processing results all the way down to the brainstem and spinal cord.
to actually drive physical behavior. And when they overlaid the functional data, the flashlight test, onto this new structural map, the alignment was just striking.
It was a perfect match.
Yeah, the PT neurons, the long-range broadcasters that preferred moving gratings, had very weak local connections in the visual cortex. But they projected heavily to a specific deep brain relay called the ledodorsal thalamic nucleus, or LD.
And meanwhile, the PT neurons that preferred checkerboards routed entirely differently.
So, to answer the question we asked earlier, yes, the biological routing physically matched the data processing type.
The brain does not use a generalized, homogenized wire to send different signals. The physical hardware is bespoke to the algorithm it is running.
Okay, let's do our second midpoint recap. We have now layered 2 dimensions. Bullet 1. We made the brain transparent and used an 8-beam laser to trace their exact wiring across the brain. That's the morphology.
So we have the job and we have the commute.
Exactly. But here's the next massive hurdle I want you, the listener, to think about. We know what the cell does, and we know its physical shape. But what genetic instructions built that specific shape?
Right, we need the blueprint. We need to sequence the RNA.
But the tissue is now a chemically cleared, hardened block of protein suspended in organic solvent. Traditional single-cell RNA sequencing requires dissociating fresh tissue, right?
Yeah, usually you have to literally dissolve the brain in enzymes to capture the RNA inside.
But if you dissolve the brain, you completely destroy the 3D structural map we just spent all this time building with the 8 lasers.
Exactly. So you don't extract the RNA, you read it exactly where it sits. They use a technique with a very long acronym, 2CE-ASI-FISH.
Okay, dual-color hairpin-encoded expansion-assisted iterative fluorescence in situ hybridization. Let's unpack that jargon step by step. First, expansion.
Right. They infuse the brain block with acrylate monomers, which are actually the exact same molecules used in superabsorbent baby diapers.
I love that visual. So these monomers penetrate the brain tissue and polymerize into a dense hydrogel mesh.
And crucially, they use chemical cross-linkers to physically anchor the RNA molecules directly to this gel matrix.
Okay, so the RNA is tied down to the diaper material. Then what?
Then they introduce water. The hydrogel physically swells, expanding isotropically.
Meaning it expands equally in all directions without distorting.
Exactly. It's like drawing a complex, highly detailed map on a deflated balloon and then blowing it up. The spatial relationships between the ink molecules remain perfectly intact, but the physical distance between them gets way bigger.
This expansion physically moves the tiny RNA molecules away from each other. It eliminates that optical crowding we talked about, allowing a standard microscope to resolve single genes that would normally be just a blurry mess.
And they mapped a panel of 30 specific marker genes inside these thick, cleared slices, doing this in 5 sequential rounds.
This is where the 2C and the fish come in. Because if you try to tag 30 different genes with 30 different colored dyes all at once, you literally run out of distinct colors on the visible spectrum.
Right, the colors overlap. So instead, they use a combinatorial hairpin-encoded approach. It's like mixing paint. They design DNA probes that bind to specific RNA targets, and these probes have hairpin structures.
Like a literal hairpin shape.
Yeah, a folded loop. And they can only be opened by specific initiator sequences. So in round 1, they flow in 2 colors, say red and green, that only bind to a specific subset of hairpins. They take a picture, they chemically extinguish the fluorescence, wash it out, and do it again.
Oh, I get it. So by analyzing the sequence of colors over 5 rounds, like if a spot glows red in round 1, green in round 2, and nothing in round 3, they create a unique spectral barcode for all 30 genes without running out of colors.
Exactly, perfectly decoding specific messenger RNAs in the thick tissue without any overlap errors.
And the findings were amazing. The transcriptomic data locked perfectly with the morphology data. Transcriptomic subtype 1, or TS1, neatly matched the long-range PT neurons, mostly in layer 5 of the cortex.
And TS2 matched the local IT neurons up in layer 23.
But there is a really cool detail here. Neuroscientists have known that IT neurons have wildly varying, almost chaotic physical shapes compared to PT neurons. And it turns out this physical variance is mirrored by high genetic variance.
Specifically, the variable expression of a gene called RORB, R-O-R-B.
Right. The physical shape actually matches the genetic chaos.
Yeah.
But we have to go even deeper. Because the IMC platform didn't just count which genes were turned on. It revealed a hidden spatial dimension within the cell itself, something called subcellular RNA localization, or sub-RNA lock.
This is the real game changer. Because the tissue was physically expanded in that hydrogel, they didn't just get a spreadsheet of gene counts. They got the exact 3D spatial coordinates of every single piece of RNA inside the neuron.
They could basically watch the logistics of the cell in action. They quantified this using 3 specific indices. First, the polarization index, or PI.
Which just measures whether the RNA is pushed or clustered asymmetrically to one side of the cell body. The gene CUX2, for example, exhibited high polarization.
Then the intranuclear index, or INI. This measures RNA that is trapped inside the nucleus, like the collagen-related gene COL6A1.
Right, it's transcribed but sequestered, not shipped out to the cytoplasm yet.
And finally, the peripheral distribution index, or PDI, which tracks RNA that is pushed all the way to the outer edges of the cell membrane.
And this sub-RNA lock acted as a whole new molecular dimension. The physical location of the RNA proved to be fiercely predictive.
Yeah, like the peripheral distribution of that COL6A1 gene actually predicted if a neuron preferred checkerboards.
Which makes profound biological sense when you think about it. Neurons aren't just spherical bags of soup. They are massive, highly compartmentalized structures.
Right, they have branches going everywhere.
Exactly. So if a neuron needs to rapidly alter a specific synapse at the far edge of its dendritic tree to process a visual input, it takes way too long to manufacture the protein in the center nucleus and ship it all the way out.
So it prepositions the RNA blueprint at the periphery, ready for local translation the exact moment the synapse fires.
It's incredible logistics.
Okay, let's do another quick recap for you listening. Bullet 1. The researchers physically expanded the brain slices like a diaper to map 30 specific genes inside those exact same neurons using the hairpin barcodes.
And bullet 2. They discovered that where RNA is localized inside the cell, the sub-RNA lock is a massive clue to what the cell actually does.
So audience interaction time. Think about this. Why do you think a cell would keep some RNA strictly locked in the nucleus, like we saw with INI, versus shipping it out to the edges? What is the cell preparing to do?
It's a question of readiness and timing.
Exactly. And now we reach the climax of the paper. We have the ultimate question. What happens when we combine function, morphology, and genetics into a single predictive model?
Well, you get a massive, highly dimensional data set, and honestly, it sounds like a data science nightmare at first.
Yeah, I mean, you have hundreds of highly complex, interrelated variables, calcium rates, projection strengths, 3D RNA coordinates, but a relatively small sample size of just 207 fully characterized neurons.
You run a huge risk of statistical overfitting. The model might just memorize the noise rather than learning the actual biological pattern.
So how do you find the genuine biological signal in all that noise?
You feed all this data into a regularized machine learning model. Specifically, they use Lasso GLM.
That's a generalized linear model with Lasso regularization. For those listening who might not be deep into ML, Lasso stands for least absolute shrinkage and selection operator. But let's explain how L1 regularization actually works.
Sure. Standard regression models try to use every single piece of data you give them. Lasso applies a mathematical penalty.
It essentially forces the model to make hard choices, right?
Exactly. If a variable, say the expression level of some random gene, isn't strongly predictive of the neuron's visual function, Lasso shrinks that variable's mathematical weight exactly to 0.
It just aggressively prunes the dead weight. It removes it from the equation entirely, leaving only the most robust, highly predictive features. And when they ran this regularized model across all 3 modalities simultaneously, the result was staggering. The combined trimodal dataset vastly outperformed any single metric on its own.
Combining RNA levels, subcellular RNA locations, and brain-wide projection strengths correctly predicted a neuron's live visual function with nearly 80% accuracy.
80%, just from looking at its form and its genetics. And by narrowing down the data this way, they discovered a major breakthrough, a brand new, highly specific, previously invisible neuronal subtype.
They named it the C2 subtype.
Let's detail C2 because this is fascinating. These are pyramidal tract, or PT neurons.
Right, the long-range broadcasters.
Genetically, they co-express the markers VGLUT1 and VIP, along with a gene called RBP4. Anatomically, they live right near the cortical midline.
And functionally, they are dual responders. They fire simultaneously for both high-contrast checkerboards and nasal word drifting gratings.
It's like finding a secret guild. It's like finding a highly specialized group of workers who share a very specific genetic stamp, live on the exact same street, and do a highly specialized job.
And given their routing and visual tuning, That job is likely related to visually driven defensive behaviors, like detecting a predator approaching across the visual field and instantly triggering evasive action.
But the hallmark of great computational biology is that it doesn't just end in a computer model, right?
No, a machine learning model is just a hypothesis until you prove it in vivo.
So they didn't stop at the math. They went back to living mice for validation. They engineered a new cohort of RBP4 Cree mouse lines,
Which allowed them to perform live calcium imaging exclusively on the neurons expressing that highly predictive RBP4 gene.
and the live functional data confirmed it perfectly. Those specific RBP4 expressing neurons demonstrated the exact strong preference for checkerboard stimuli that the trimodal model said they would.
They proved that their trimodal model works in the real world. The correlations found in the cleared dead tissue accurately dictated the living biological reality.
Which brings us to the final synthesis. The IMC platform successfully linked live behavior, whole brain wiring, and 3D molecular blueprints within the exact same single neurons. It literally moves neuroscience from 2D flat maps to a fully integrated, multidimensional understanding of the brain.
And why should anyone care? Because this isn't just about mice looking at checkerboards on a screen.
No, not at all.
This platform is a ground truth resource. It gives us a template for how to decode the biological hardware of any thought, any behavior, or any disease in the future.
Yeah, think about Alzheimer's or Parkinson's. We no longer have to guess how a genetic mutation alters a cell's physical wiring and how that degraded wiring messes up cognitive function. We can measure all three domains simultaneously, tracking the pathology from the DNA straight up to the behavior.
It is truly revolutionary. And, I want to leave the listener with a final provocative thought, a biological puzzle to mull over.
Oh, I love these. Let's hear it.
We saw how crucial the physical location of RNA is to predicting what a neuron does, the sub-RNA lock. And we know that learning and memory require the rapid synthesis of new proteins at specific synapses to strengthen connections.
Okay, I'm following.
So if the brain prepositions RNA at the periphery of the cell to be ready for these inputs, could our daily experiences and learning actually be physically shuffling RNA around inside our brain cells as we sleep?
Wait, really? Like physically moving the blueprints during sleep?
Exactly. As we sleep and consolidate memories, is the brain physically migrating RNA transcripts across the dendritic branches, repositioning the molecular hardware for the next day's experiences?
Oh, wow. So you're asking, are our memories fundamentally stored not just in the connections between the cells, but in the exact 3D geographic coordinates of RNA waiting at the edges of our synapses?
Precisely. What does that mean for how our memories are physically stored and retrieved?
That completely reframes how we think about the physical weight of an experience. The brain isn't just changing its wiring, it's constantly dynamically migrating its genetic blueprints to meet the demands of the world.
It's a beautiful system.
It really is. Well, thank you for joining us on this deep dive into the multidimensional architecture of the mind. Take that puzzle about memory and sleep with you. Keep questioning the world around you, and we will see you next time.